Hello, everyone, and welcome to today's webinar titled Accelerate Your Immunological Research with High-Speed Clog-Resistant Spectral Flow Cytometry. This webinar is a part of the Immuno-Oncology virtual event series. I am Sydney Pinilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by Thermo Fisher Scientific. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. Okay, now let's go ahead and get started. I now present today's speaker, Eric Finan, Senior Manager, Systems Design Engineering, Thermo Fisher Scientific. For a complete biography on our speaker, please find it to the left of your screen. Eric, you may now begin your presentation.
Thank you for the introduction. I'm Eric Finan. I'm a Senior Manager in Research and Development in Systems Design Engineering and Flow Cytometry in Fort Collins, Colorado for Thermo Fisher Scientific. Thanks for joining me today. I'm really excited to talk to you about how you can speed up your research with high-speed clog-resistant spectral flow cytometry. So today I'm going to talk about a few different things. I'm going to talk over high throughput advantages for immunological research. We're going to talk a little bit about acoustic focusing technology and its impact on data quality. We'll talk about clog-resistant design and maintaining workflow efficiency. And then I'll close out with some practical insights and tips for minimizing clogging and improving productivity in your lab. So what are some common flow cytometry issues? Especially if you're working with immunology or immuno-oncology samples, they can be really clumpy. They've got these high cell densities or debris. They can have these chunks in them and can easily lead to these blockages, as shown on this slide on the right here. These plots may look familiar to you, unfortunately familiar, where maybe you're running a really challenging sample and you see these kind of gaps, these like micro gaps or micro clogs in your sample. You know, these clogs can not only interrupt data acquisition, but can also lead to sample loss and instrument downtime and can increase maintenance efforts. So let's say you get like a full clog. Maybe, you know, you have to get out a syringe or undo some tubing and get in and try to actually clog the instrument. It's sort of challenging to get through those samples and really extends the length of your experiment, of your data collection. So a cytometer that's designed with clog-resistant features to handle these really challenging samples can really help provide consistent performance and higher data fidelity. So I want to talk about what sets the Attune family of cytometers apart. All in-featured Attune flow cytometers feature acoustic focusing and syringe-driven sample. This acoustic focusing gives us gentle focusing of cells in space. It enables higher sample injection rates. It reduces our clogging because we have that syringe-driven system, and that syringe-driven system gives us absolute counting capabilities. All of the Attune cytometers feature spatially separated flat top lasers. So if we look here on the right, we can see on the in the middle of the plot here a Gaussian-shaped beam and our particle, and we can see that it's a little bit misaligned with the most intense part of that Gaussian beam. But if we have a flat top laser as shown on the bottom right here, that particle can move a little bit in our sample core, but still get hit with the same number of photons or the same intensity of photons, which means that we get the same intensity of signal and across all of our particles. And so that can actually reduce our CVs. All Attune cytometers also feature high-through capabilities. So they're compatible with an autosampler that runs 96 and 384 well plates and also goes with lab automation equipment. So I'm going to talk a little more about acoustic focusing or acoustic assisted hydrodynamic focusing. On this slide is a really cool series of photos from a glass capillary with, on the left here, no acoustic focusing. So we're looking at our sample, we've got all these particles in there, they're spatially distributed in our capillary, and we're pushing that up towards our interrogation region. In the middle photo, if we turn our acoustic focusing on, you can see that that acoustic field actually encourages all those particles to go to the center of our capillary. So they're well aligned in the middle of the capillary. And on the right here, if we were to dilute that sample, we can see that we're starting to see that sort of single file line of cells or particles that we're really interested in. And that is really what we're after, because as those particles then transit up towards the laser, we get the analysis cell by cell by cell. And it's really that single cell analysis that we're after and reducing our coincidence rate. So here we really want to be able to have our particles well aligned and in this single file line. So why acoustic focusing? This is a really cool video on this slide. This is actually looking down, right down the barrel of the capillary, if you will. And you can see that we have this spatial distribution of the particles. And when that acoustic focusing turns on, it focuses it to a very tight line right in the middle of that capillary. This enables precise alignment of the cells with the laser of interrogation. This means consistent and reproducible data with lower CVs. And this enables higher flow rates without sacrificing data quality. So this is what's so great. It reduces your time to obtain statistically significant data. So the Attune Xenith Flow Cytometer offers flow rates of 12.5 all the way up to 500 microliters a minute. Later this year, we'll be enabling 1,000 microliters a minute. And this reduces your time to record controls and samples. So if we look at the table on the bottom right of this slide, for instance, under a typical hydrodynamic focusing only regime of 60 microliters a minute, in this experiment, it took about an hour to get to 1 million granulocytes. But with acoustic focusing at 200 microliters a minute, we can get that same data with increased precision in less than 15 minutes. At 500 microliters a minute, we can do it in less than 6 minutes, and 1,000 microliters a minute, again, with the same or better precision in just over 3 minutes. When we think about moving to larger panels in flow cytometry, especially spectral flow cytometry, we can expect we'll be doing more work analyzing rare populations. So the rarer the population, the larger number of overall cells you need to analyze to get to statistically significant data in those rare populations. Additionally, the bigger the panel, the larger the number of controls that you will need to record. So increased speed means less time to record all of those controls and to get to that statistically significant data. So we can really see the power of getting to the data you really care about much faster with acoustic focusing on the Attune Xenith. Additionally, we feature specific volumetric delivery. This makes it really clog resistant, even for challenging sample types, and it provides consistent concentration results. So we're going to take another look at acoustic focusing by the side and break it down about how this promotes very good CVs or very great precision. So on the left here with low sample rates, 10 to 20 microliters a minute, if we're all the way on the left with traditional hydrodynamic focusing, we can see here that we have a very low sample input rate and we can have a very high sheath rate. And that hydrodynamic focusing will then make that core stream or that sample stream very thin as it goes in, or very narrow as it goes into the laser region for interrogation. This very small spatial distribution means that it hits the same part of the laser and results in this very narrow distribution of intensity in your histogram. Now, if we were to increase our sample rate and just maintain traditional hydrodynamic focusing, that means that as a proportion, your sample core is going to be a larger proportion of that mass flow if the sheath rate remains the same. And so your sample core actually grows in diameter, as we see here in this third photo. And so with that, as that core diameter grows, we're hitting many different parts of that laser, and we can be collecting different intensities of light, because no lasers are perfect. All lasers have a little bit of ripple on them, even if it's a flat top. And so what we end up with is this wider distribution of the signal of what we're after. And that makes spectral unmixing a little more challenging. We start to see some artifacts from that. Now, if we look at acoustic assisted hydrodynamic focusing at this low sample input rate, we can see that, the sample core is already fairly narrow. And so the acoustics really don't make a tremendous difference here because we already have a pretty narrow spatial distribution in our region of interrogation. And so we see about the same result. But where the power really is here is in these higher sample input rates. And so when we turn on the acoustics and we can focus those cells to the middle of the capillary before we hydrodynamically focus, then we get this really great tight alignment of those cells in the center of that laser. And so with this, we can have a really narrow distribution even at our high sample input rates. So what does this actually look like in an instrument? Well, here's some example data that was collected on the Attune Xenith, and we can see that the acoustic focusing allows faster acquisition without compromising resolution, and that the stain index is maintained across our flow rates. So we can see here in the middle, we've got APC, BUV 395, and BV 785 run at 100 and 500 microliters a minute. Those are bolded in the table off to the right. And we can see that virtually they look identical. They're very similar across these different flow rates, indicating that we can maintain that precision across our different sample input rates. So let's talk a little bit about clog resistance in the Attune Xenith. So as I mentioned, I'm an engineer in the intro, right downtown Fort Collins, Colorado, and decided, hey, I'd go out and buy a very common sample for an engineer, not so common for an immunologist. So you can see here, I actually went out and got some orange juice and I put it on the instrument and I thought, okay, what can we do with this instrument? Can we really push it beyond its limit? So in this first example, I did filter the orange juice down with a 70 micron filter one time, and I diluted it to 20% concentration with DI water. And then I ran it at all the sample input rates that we offer on the Attune Xenith. And what this goes to demonstrate is that we actually have great data quality and consistency at all of these different sample input rates. This table on the left also highlights that we have at this enormous dynamic range that we can go anything from 1,000 events per second and all the way up to at 1,000, we're showing just shy of 100,000 events per second. Now, of course, that's gonna be riddled with coincident effects. And so, maybe not a great event rate to be working with at the highest end. But again, this is just an example. You should not be running orange juice in your lab. But what this goes to show is that we can acquire data faster without compromising on the quality. So I want to highlight part of the clog resistance that's available on the Attune line of cytometers, including the Attune Xenith. So how does this really work? Well, having a syringe-driven sample means that we maintain consistent volumetric flow and not pressure. And so this consistent volumetric flow, as you see here in these three graphics, on the bottom left, under normal flow, we're running at about 7 PSI in the region of interrogation. But let's say you're working with a really challenging sample. You can see here in this middle photo, maybe we've got this big clump that comes up into it and provides maybe like a micro clog, if you will, or a partial clog in the instrument. Well, because we're maintaining consistent volumetric flow, the pressure will actually increase to maintain that flow, as we see here on the right, and that overpressure will displace that clog, will push that through and continue to flow in the instrument. And so that's really the power of having a syringe-driven sample, is that we can continue to maintain that flow even when you have differential back pressure. So what would it look like then if we really challenged the system? What happened if we just put in orange juice neat? What if we didn't filter it at all? As in this photo here, just right into the tube. Well, for reference, I put a few drops of this orange juice, my sample, onto an EVOS M5000 and took a few photos. And you can see here the scale bar is at 1 millimeter or 1,000 microns. And you can see here that we got some really big chunks of pulp in this orange juice, ranging from 500 microns up to more than 2,000 microns. And for reference, our flow channel is 200 by 200 microns square. So a more than 2,000 micron chunk of pulp needs to squeeze its way through that flow channel. So what might that look like on the Attune Xenith? Now, I'm going to flip to this slide, but I do want to warn, please don't try this at home or in your lab. This is run by an engineer in an engineering lab, and I can handle anything that could have gone awry. But we can see here at these different flow rates that the Attune Xenith actually pushes through these partial clogs, even with really challenging samples. We see this and no, this data isn't great. You would certainly want to publish this data. We do have a lot of gaps. It doesn't really look great. We can see here actually on the bottom right that I got notified of an actual clog at our highest flow rate. But again, this is a really, really challenging sample to push through the cytometer. And we can see here that mainly it handled it well. We can also see here on the bottom left, the spectrum of orange juice doesn't look like it would make a great floor, but it is kind of neat to see. Okay, so what happens if you do get a clog? Well, we've got a great workflow here where we actually use a pressure sensor to detect that clog. We present to the user, would you like to recover your unanalyzed sample? And you can either use the same tube or put a new tube on there, recover your sample back that's unanalyzed. Then you can run unclog with one click, hands-free from the instrument. That unclog will run, it will automatically rinse the instrument, and then you can continue running with an append workflow if you want to put that back to your previous sample or with a new sample. We recognize that this is actually really powerful and really helpful for core labs or labs where there are a lot of different users with different challenging samples, as it doesn't require an expert to run this. Both you can run really challenging samples, but if you do get a clog, we sense it, we provide right to the user, hey, you can go ahead and run this unclog, and then you can continue running. We're able to do this because we have this high pressure unclog built into the instrument. So you can see here on the right that we actually use the syringe pump that we have to push backwards through our interrogation region, and we push any clog that might be in the region of interrogation to waste. Okay, so as exciting as orange juice might be to you all, you might be more interested in a mouse lung panel or a real panel here. So what we have here is a basic 14-color mouse panel looking at populations in the lymphocyte, monocyte, and granulocyte populations. These were harvested lungs that were digested using collagenase at 37 degrees C and then processed once through a 70-micron filter. Here, we're looking at several populations of interest, including CD4 and CD8 T cells, alveolar, macrophages, and neutrophils. With this backbone of a panel, we hope to enable customers to perform high-speed analysis of their more particular markers of interest on lung cell populations. And what's shown here was collected at 100 microliters a minute. So I'm going to go to this next slide and show running this at different sample rates. So with that single 70 micron filtration, we got about 3 × 10⁶ per mil of concentration, and we can see that it's actually pretty challenging. So here this shows up on our first time plot that as we're trying to run it, at that concentration, it is kind of clumpy, and we see that in the gaps of the data here. So while we do have fairly stable scatter intensity, this really doesn't look that great, and it might be challenging to analyze, especially on from a time dimension. So what about if we just dilute it and run it at a lower event rate? So this is the second condition. We dilute it down to about 6.6 times e to the 5. We're getting an event rate of about 1,000 per second. And we can get the same data, but if we run it at that same flow rate, it's now going to take us 5 times longer because we diluted it 5x. It's going to take us 5 minutes to get this data. Now, the power of the Attune Xenith Flow Cytometer is we can actually just turn up the sample acquisition rate. So we can run it at 500. So at the same dilution, 6.6 times e to the 5, we get our event rate up to just north of 7,000 events per second. And we can see here that the data looks really great. It's really stable. Let's say that we were still encountering some challenges, or we just wanted to collect our data faster. We could dilute it some more. We could dilute it to 10x as shown in this last plot and actually run at 1,000. And again, we can see here that we get really great stable data across this range. And what this really highlights is that dilution is a great way to make sample prep easier. We don't have to filter multiple times. We don't have to wait on that. We don't have to do anything else. We can just dilute it and just run faster. And that's really the power of the Attune Xenith. So you might be asking, okay, so what does this data actually look like? What does the panel actually look like across these different flow rates? Well, I'm happy to show on this slide that, this data collected at 100, 500, and 1,000 essentially looks identical. And really what this shows is the power of the precision of the acoustics that we have, especially at these higher flow rates. And so, it really is great. We can, reduce our time to get to that, the data that we're really after, that statistically significant data, dramatically by running at these faster flow rates without compromising on data quality. So I'm just going to finish up here with some recommendations on these challenging samples. I've sort of walked through them a little bit, but sample prep must be really carefully considered when working with tissue digests. Always filter your samples. Unlike my orange juice example, always filter your samples. And particularly when working with poor quality samples or difficult tissue or tumor samples, quality control checks are essential. And so what's shown here on the right are a series of acquisitions from a clog-prone tissue digest that was actually prepared from a whole spleen. And looking at those time plots really is a great quality check to understand how is my sample prep, how is it actually being acquired in the instrument, really important to check on that. And then as just highlighted, diluting your samples and increasing your sample input rate can reduce your clogging and increase sample stability without sacrificing any precision. So this is all available on the Attune Xenith Flow Cytometer. It has the acoustic focusing designed for efficiency, speed, and accuracy. We have high resolution, spectral, and conventional compensation workflows, and it pairs up with the Invitrogen Sidekick Max Auto Sampler. It is the fastest spectral flow cytometer on the market. The acoustic focusing allows it to run 5 times faster than most spectral cytometers. It's really great with sticky samples. We've got reduced clogging as shown in this presentation. Really helps with detecting rare event populations. Again, that statistically significant rare population is really enabled by being able to run faster. And then it pairs with the Sidekick Max that has 96 and 3/4 well plate acquisition compatibility. So I didn't even highlight it, but just to mention it, we have 6 lasers with 51 fluorescent detectors and 6 scatter channels. We have enhanced diagnostics, so we have onboard service cameras that really provide great remote support. I'm actually one of the engineers who works with our field service team in case there are any issues, and having these onboard cameras makes remote troubleshooting really fast and easy, which minimizes any downtime for our users, maximizes their uptime and data collection. And again, it is equipped with both spectral unmixing and conventional compensation workflows. So thanks for joining me today. I really appreciate it.
All right. Thank you very much, Eric, for your informative presentation. We will now go ahead and jump into the Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just submit the question in the Ask a Question box located at the bottom of your screen. Okay, let's go ahead and get started here. And just so everybody is aware, we have our speaker connected via audio, but there is no webcam video for this portion, for this Q&A portion, just to avoid any confusion. Okay, Eric, so the first question we have here is, does acoustic focusing maintain robust signal resolution for small or dimly labeled populations?
Thanks. Yeah, that's a great question. So the acoustic focusing doesn't change anything about the level of fluorescence in your samples and any of your populations. Really, as just overviewed, it helps center those cells of interest or those particles in the same place as a laser for interrogation. So what this does is it reduces CVs or increases the precision of the data. So this can help in resolving the difference between populations that have similar levels of fluorescence. If you think about 2 bell curves that have much narrower distribution, you'll be able to look at the medians, the difference between the medians, if they have narrower distributions much more easily. But it doesn't change the median intensity of a population.
Got it. Thank you. Okay, let's take a look at the second question we have here. It says, what maintenance protocols are recommended to keep the acoustic focusing system running optimally?
Got it. Yeah, thanks. Yeah, so no maintenance is required to keep the acoustic focusing system running well. At install, a field service engineer will verify the calibration of the acoustic focusing capillary that we use. That is actually calibrated and set in manufacturing, and a field service engineer will just verify that things are working well, maybe tweak a couple of powers if needed at install to make sure they're optimized. But beyond that, no additional efforts required, and it continues to work well.
Wonderful. Thank you. The next question we have here says, what software tools are available with the Attune Xenith?
So there is a software that's being launched with the Attune Xenith. If there is anyone out there who is current Attune users, you're familiar with the Attune Cytometry software. But with the Attune Xenith, we're actually launching a software that's based on SQ software, which is the Sasquatch software that runs the Bigfoot cell sorter. That's used to both drive the system and acquire data. It will feel very familiar to both Bigfoot cell sorter users and Attune Cytometry software users. It's designed to be able to switch back and forth between both the Bigfoot cell sorter and the Attune Xenith with the same workflows, to make it much easier to go back and forth between these two fairly compatible instruments.
Perfect. Next question. Is the Attune Xenith available in 3, 4, and 5 laser options?
Yeah, it's a great question. We currently are offering it in one configuration, which is the 6 laser option. We chose these lasers and the detector layout to best suit the needs for immunologists and immuno-oncologists.
All right, perfect. It looks like we have one final question here. If you are in the audience and happen to have a question you would like to ask, I encourage you to submit that now. If not, we will go ahead and jump into the last question here. It says, does Xenith have its own analysis software, or do users need to use third-party analysis software?
Yeah, fairly related to a previous question. Thanks. The Attune Xenith does come with its own acquisition software. As I mentioned, that's currently called SQ. If you are a Bigfoot cell sorter user currently, it will look like that, but there are some substantial, I would say, upgrades that make it much more compatible with a flow analyzer and much easier to manage many more samples. We also have updated our workflows in that for both conventional compensation and spectral and mixing. And this updated software does come with many of the analysis tools that's needed for both conventional and spectral analysis. It is compatible with third-party analysis software, but a lot of the tools needed for basic analysis are already included in that software package.
Perfect. Thank you again, Eric, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we did not have time for and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2026. LabRoots will go ahead and alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event.